So just sort of a very big picture, and I think that should be pretty obvious. To do science, we need data, information, and analyze it in the right form. And there are different ways we can get data, and there's sort of different types of quality of data. If you're doing experimental studies, and for immunology, that is where most of the data comes from. You have a lot of control. Especially if you use animals, you can measure a lot of things. So even they are not everything. Sometimes we would like to measure things we can't access, at least not at the level of detail we'd be interested. But in general, and with experimental studies, you have the most power and most control. But of course, we know we can't always do that. We can do certain things with certain animals, but when it comes to human studies, we're much more limited. Then the next level, and that's where public health and other fields, certain areas of medicine too, and then of course other disciplines get lots of their data from, sometimes all of their data, which is observational studies. So you can't sort of manipulate the system, but you can carefully observe the system, and hopefully by analyzing the data properly, you can still draw some useful conclusions about what's going on. That cartoon kind of illustrates the problem we sometimes have, that you might see spurious correlations, but they might or might not be causal, and therefore the next study that comes around might suggest that no, actually the opposite is true. So that's just a limitation with these kind of studies. And then the simulation and modeling studies, at least the kinds we will be talking about, you can think of another form of generating data. You try to build a model that approximates the real system, you run simulations, and you can generate data from your model. It's of course not as good as real data. If we can get real data, it would be good, but sometimes that's our only option. And sometimes even if it's not the only option, it might be a good supplement to collecting actual data. And so those are the kinds of models we'll be talking about here. Just a bit of terminology. It's maybe not quite as bad as immunology, but not that far away in terms of the definition of terms or how fuzzily they are used. When we talk about modeling in science, we usually mean some kind of math or computational models, but that itself is very broad. You can have all kinds of models there. We're talking specifically about a type of model that I'll define more closely in a few slides. But in general, it's some kind of mathematical or computational, or often both. You start writing it down mathematically and then implement it on a computer. So within sort of the big bucket of models, there are lots of different ways you could classify and categorize models. Are slides larger? Okay, let me see. Is this better? How do the slides?
Yeah, that looks a lot better.
Okay. Okay. And you also have the slides if you want to annotate them or so. You can download them and make comments on them. So this is a way you can classify models. Of course, not the only one, but I think it's helpful, especially since a lot of you might be familiar with the first kind of models that are classically called statistical models. I don't necessarily like the term too much because the models we'll be talking about can also be used in sort of a statistically model fitting sense. I like to call them phenomenological models. And so those are the big class of models that are used to, broadly speaking, look for patterns in the data. They don't describe the mechanisms that might have led to the data, but they're looking for overall patterns. And you can contrast them to the kind of models we'll be talking about, mechanistic or simulation models. I use the term interchangeably. They're trying to explicitly represent mechanisms. And as you'll be seeing, you can use them with and without data, while the first kind of models you always apply to data. So these kind of what I call phenomenological models, you might probably all familiar with. Even if you haven't done too much stats, you probably have done a linear regression or at least a t-test or something when you had data. So what you do with those, you use some kind of mathematical formulation of a model to try to see is there a pattern in the data. So in a linear regression, you see is there a trend of one variable, as you plot it as a function of the other variable. Nowadays, this field has expanded a lot with more data available. The methods we can use can be more complex, and we can still get interesting things out of that. So the buzzwords these days are machine learning and artificial intelligence and deep learning. They all fall into that big class that you use some potentially complex statistical model to try to get information out of your data, basically finding patterns in the data. And so most of these models don't actually think that the way you built the model incorporates a mechanism. So you're just saying, is there a linear pattern or is there a curved pattern in the data? But you don't necessarily think that there's necessarily a one-to-one mapping to a mechanism that leads from from a certain variable to some outcome. So here we really want to understand, is there patterns in the data? Is there something, with like statistical significance? Is there some trend in there? And then potentially, if the models are good, we can try to make predictions. Here's an example. Paul actually mentioned that earlier in his talk. A paper we did a few years ago, last year came out. We looked at T cells and different epitopes and looked at what led to the different strengths and responses among these epitopes, and looked at certain predictors such as the amount of MHC that is directly presented and cross-presented, and protein levels. And so this is just a standard regression model fit to the data. You see there in the figure linear regression, trying to look at these different variables on the x-axis with T cell response on the y-axis. So I'm sure you've all seen those kind of figures, and probably most of you have even done these kind of analysis. So that's useful because we can see, okay, is there a correlation between one variable and another one? And we don't actually have to know in very detail the mechanisms that give rise to this kind of pattern. So finding these correlations pattern is relatively simple. You still have to do it right, and statistics is not easy, and picking the right model and doing the right interpretation is its own big topic. But in general, you can sort of, at least if you use simple models, sort of use out of the box. You can use a linear model, a logistic model, and further additions to that. And it's not that hard to apply them. And often these models are very useful. Definitely you can find correlations, and then some of these models are very good at predicting. So, a lot of stuff on the internet is driven by good statistical models. Netflix tries to tell you what movie you should watch next. Google Translate is using models to try to, not only translate words, but translate whole sentences and try to get the right way to translate a word in a specific context. And while narrowly these kind of statistical models look at correlation, depending on how the data is collected, you might go from correlation to causation. So if we run a clinical trial or we do a lab experiment and the only thing we change is a variable we're interested in, we can look at differences, and then we can say, well, this was the only thing we changed, so we think this thing actually caused the outcome. When you have observational studies, that's a little bit trickier. But if you do a careful analysis, you might still be able to say, okay, this might have caused that, which is what we care in the end. Correlation is really, you can find correlations anywhere, but it's only the first step. What we usually care is one thing affects the others in some way. And the good thing is we can do all of that without really having to understand the mechanisms by which one factor affects another one. So we can just see that and say, okay, this affects that. So an example I sometimes like to make is, it's completely clear that, smoking leads to an increased risk in cancer. And we might not even fully understand it at the biomolecular level completely how that happens, but it doesn't prevent us from saying, yes, the evidence is so clear that we should try to get people to not smoke. And there are lots of other examples like that, where we might not fully understand all the mechanisms, but the data is so strong. There's probably, I don't know what fraction, but a lot of drugs we might not fully understand how they actually act in every detail, but the evidence is clear that they work, and therefore we should use them. So you can get very far and actually make actionable, get to actionable insights without having to understand all the mechanisms. Of course, the flip side of that, the disadvantage is that sometimes if you try to go from correlation to causation, you might stumble. And sometimes a jump is not justified. And that's where we get these studies that report that X causes Y, and then a year later someone reports, no, that's actually not the case. And the other thing is, even if it's okay and the causal relation is there, you don't really understand the mechanisms based on these models. You just see, okay, the data, the outcome, and the predictor are linearly related, or there's a sigmoidal relationship, but you don't know how, in terms of mechanism, one maps to the other. So those are the disadvantages of those kind of models. And now the flip side of these are these models we'll be talking about in this class, these mechanistic models. So the idea behind is that you take a system and you try to build the mechanisms or processes that are going on in the system, of course, in very simplified forms. And you'll see that throughout this course, our models are incredibly simple compared to what you just learn, how complex the immune response is, and everything else Paul will tell you. So there's always this tension about having a very complex system and trying to make simple models that are still hopefully useful. So you take a system and you formulate a model, usually with mathematical equations. Doesn't have to be, can also just be computer rules. Almost always these days at the end, even if we start out with equations, we implement it on the computer and then explore it. Again, terminology, I'm going to switch around between mechanistic and simulation models, but you will find all kinds of terminology. Sometimes mathematical models, so that's a bit of a misnomer because statistical models are also mathematical. Dynamical models is more in the engineering field. ODE models, which is not quite right, but most of these models are actually differential equations, as we'll discuss. So again, terminology is kind of fuzzy, but usually when you see it, you know if you have a mechanistic model or not, and you'll know that by seeing that there's the actual processes going on in the system are in the model. And we'll discuss that in more detail. You can see in that figure, I already pulled one up, you see certain processes of a viral infection being implemented, and we'll discuss through that in detail. It's just a quick example I pulled from an old paper of mine. It's T-cell responses, and we looked at the immunodominance question that Paul mentioned. And we have 4 equations, and we have various processes here implemented in the model. So you have here a process of virus growing, you have a process of the virus being cleared by the T cells, a process of naive T cells being activated, and so on. So very simplified what's going on in the real system, but explicit processes or mechanisms. And that's why we call it a mechanistic model. What you gain from those models is that you potentially can understand the system in a deeper way, because you're now implementing the mechanisms and how things interact. And by exploring those, perturbing them in your model, you can understand how a certain mechanism affects the outcome. And you really know how the different components interact with each other and then affect the outcome. So in this example, if I wanted to know how that growth rate R of the immune response here affects the outcome, well, I can study that in my model in a lot of detail. The disadvantages are that, to build these kind of mechanistic models, you actually need to know, or at least assume, something about your system. So you need to be able to say, okay, these components are important, and this is how I think the components interact with each other. So if you don't have any knowledge about your system, usually when you're new and study a system for the first time, you'll probably first want to do sort of the statistical modeling approach, the phenomenological one. And then once you have more sort of understanding of your system, maybe these mechanistic models might be a useful tool. Of course, if you build a model based on the wrong assumptions, then any insight you gain from your model will not be very useful. If the model is wrong, then what you learn from it is also wrong. And so that's, of course, the tricky part with these kind of models. So just to summarize these non-mechanistic models that you've probably all seen and used in some form, you use them always with data. You're trying to find patterns in the data, looking for correlations, and then maybe be able to go to causation and prediction. And you don't really need to understand in details how the data arose, by what mechanisms. On the other hand, these mechanistic models, are useful when you want to sort of zoom in, go deeper, and try to study mechanisms by which you can certain patterns. And I hope from just a few slides I mentioned, you realize there's no one is better than the other. It very much depends on your question. So ideally, you have both in your toolbox, and depending on what the question is, you pull out the right one to address the question for which one or the other model is more suitable. And often within a single project, you might even do both. You might start with one kind of model and then move on to the other kind. So it's just the kind of models we'll be doing in this class. So we'll focus just on these mechanistic simulation models. And the important part is that the processes that we're interested in that happen in the system are in the model explicitly. And we specify them, and then we can play around with them and explore them. Just to make it a bit broader, outside of immunology, these models are used in a lot of fields, especially fields where things are man-made. Engineering is a lot. Weather forecasting, so we kind of know the equations that drive the weather, so we can simulate them on computers and try to predict the weather. In economy, you can try to build model. Then infectious disease, some of you, I think, work on the population level. They have a much longer history on that level, and I'll have a few slides on that. And then over the last decades or so, more and more immune response modeling. Basically, any field where you have a system where you know enough about it to write down or specify some components and processes where you can potentially use these kind of models. Here's a quick example. We'll see that later. This is from a 2001 paper in Journal of Immunology. So what the authors did, they built a model, and then they ran the model. It's for TB, and they were interested in trying to figure out what would perturbation of certain cytokines that are considered important for TB infections do to the system. So as you can see here, for all these four quantities, they track interferon, macrophages, total bacteria, and T cells. They let the model run to get a steady state. That's basically mimics a chronic infection of TB. And then basically with their model explored what if they perturb these components of the model. So the model had IL-10, IL-4, IL-12 in it. And these are different cytokines. And so in the model, because you have full control, you can perturb them, turn them on, turn them off, and see what happens. And so then that's basically what they're doing here at around day 1000. They turn them on or turn them off. I think in this case, they turned them all off. They depleted them. And then you can see what the outcome is. And so you see in this example here, if you turn off IL-10 and IL-12, bacteria load goes through the roof. Basically, that would mean you get you move on from the chronic stage in TB to the disease stage. While for the other ones, it looks like it actually reduces bacterial burden. So if we believe that this model is an adequate representation of the real system, and that is always a big if, and that always needs to be checked, and we'll mention that more than once and get to that, then you could say, well, this is what we predict what would happen if we could perturb those cytokines. And ideally, now you might want to do an experiment to see if you can do that. But it might be something that you can't actually currently study. So then you could use a model to say, okay, we think this would happen, and then that might be suggesting that this is a useful venue for further research to try to be able to perturb these kind of cytokines. Here's another real-world example from the world of infectious diseases on the population level. You can find tons of these kind of models currently for COVID, and I'm sure you've seen some of them. All of them are these kind of simulation models that most of them try to implement the processes of individuals infecting others and how it then spreads through the population. This one was for a potential influenza pandemic, and obviously we can't collect data because the pandemic at that point hadn't happened yet. It happened in 2009. Fortunately, it wasn't too bad. But so they were basically running their simulation models, looking at what would certain interventions do. So on the left column, you see baseline. You don't do anything. You see red, a lot of people get infected, and then it dies down. And on the right, you see prophylaxis with antivirals, and you see it's delayed and somewhat reduced. So those are the kind of things you can do with these models. Run simulations, try to predict what would happen. Again, if you believe that this is a reasonable representation of the real system of reality, then you can actually use that to make decisions. Just a few slides putting the two next to each other, within host modeling and between host modeling. We'll focus here on within host modeling, but I mean, in epi department, I do a lot of between host work as well. And more importantly, it's been used in that field for much longer. So a lot of the approaches and methods that we'll be talking about were actually first developed on the population level. So you can often find good insights and approaches and ideas in that literature. The equivalent is basically on one, you model the spread in the population, and the other one, you model the spread inside a host. Other than that, again, methodologically very similar. So just keep that in mind. As I mentioned, population level modeling, much older, several hundred years now with Bernoulli. It's actually a very nice paper if you're interested in that reading. Sally Blower sort of republished the paper with commentary in 2004. So you can take a look there. And then definitely over the last 100+ years, a good bit of activity. Within host modeling, much more recent, kind of started sort of in the 70s, 80s, but since then has seen a lot of rapid increase, both in terms of how sophisticated the models are and the quality of the data. So it's definitely been catching up quickly with the population level modeling. And that's all I had on the general overview of these kind of models. Let me pause here and ask if there are any questions. I can see the chat window, so you can write them in there, or you can raise your hand and ask them. And then after that, we'll move on to the few small model introduction.